Welcome to episode 7 of the Physiology 225 podcast. I'm your host, Dr. Jim Davis, and today we're tackling the fascinating topic of transport mechanisms. These processes are critical for understanding how materials move into and out of cells to maintain homeostasis. As always, we'll keep things simple and approachable, and remember, in our next episode, we'll dive into the topic of membrane potential. For now, let's get started. So the first question we need to ask ourselves is, what do we mean and what are transport mechanisms? Well, transport mechanisms are the way materials like ions, water, and molecules travel across the cell membrane. These processes are essential for maintaining the balance of intracellular and extracellular environments. The primary types of transport mechanisms include simple diffusion, facilitated diffusion, active transport, exocytosis, and osmosis. Now, this is not a comprehensive list, and there are some things like endocytosis that would be part of this, but we're not going to talk about them because we won't talk about that at all this semester. So to start off, let's talk about simple and facilitated diffusion. Simple diffusion is the movement of molecules directly through the membrane. It's driven by a concentration gradient. Molecules move from an area of high concentration to low concentration. Just want to remind you that I told you that flow down gradients is involved in a lot of things. For example, oxygen and carbon dioxide diffuse freely across the membrane. Facilitated diffusion, on the other hand, requires the help of proteins like channels or carriers to move molecules across the membrane. It's still driven by a concentration gradient and doesn't require any outside energy. Think of it like needing a key to unlock a door. Next, we have active transport. And active transport is like swimming upstream. It moves molecules against their concentration gradient from an area of low concentration to high concentration. This process requires energy, usually in the form of ATP, and also involves proteins embedded in the membrane. A prime example is the sodium-potassium ATPase pump, which helps maintain the balance of sodium and potassium ions inside and outside of the cell. And we will talk a lot about that as we go throughout the nervous system. Exocytosis is the process of moving large molecules out of the cell. Molecules are packaged into containers that are called vesicles. These vesicles will fuse with the membrane and release their contents into the extracellular fluid. This process requires ATP and is crucial for functions like hormone release and neurotransmitter signaling. So when we talk about a chemical synapse, we're going to come back to exocytosis. Finally, we have osmosis. Now, osmosis is a special kind of diffusion. It's the movement of water across a semi-permeable membrane, and that means that only certain things can cross the membrane. This movement of water is driven by differences in solute concentration, and solute concentration is also known as osmolarity. Water moves to the area with a high concentration of solutes such as salt. Osmosis doesn't require energy because it's powered by the concentration gradient. Basically, what happens is salts or proteins or something else is attracting the water. And so if you have a lot of salt in one area and not so much in another area, those large number of salt molecules will attract water. Now, while osmolarity refers to the concentration of solutes in a solution, there's something called tonicity. And tonicity compares the osmolarity of two solutions and describes the movement of water. So we've got three different types of tonicity. We've got hypertonic, isotonic, and hypotonic. If we have a hypertonic solution, this means that there is a higher osmolarity in the fluid outside of the cell than inside the cell itself. This increased osmolarity causes water to move out of the cell, and the cell shrinks, or it has a cool name, crenate. Next, we have an isotonic solution. This is where we have an equal osmolarity inside and outside of the cell. Ultimately, this means that there is no net movement of water, and the cell will stay the same size. Now, in all of these cases, water is moving in both directions, but it's just a matter of which direction is water moving in more frequently. In an isotonic solution, for every molecule that goes in, you have one molecule that's going out. Finally, we have hypotonic solution. Now, in a hypotonic solution, there's a lower osmolarity in the fluid outside of the cell, which causes water to move inside of the cell. And so the cell will swell, or it might burst, or what we call lyse. Finally, I want to end with talking about the primary ions that we talk about with the cell membrane. And so the distribution of ions across the cell membrane is vital for the cell function. And we'll get into that more a little bit later in future episodes. So we have potassium, and potassium is found in much higher concentrations inside the cell. It is a positively charged ion. Then we've got sodium, which is also a positively charged ion, but it is found in much greater concentrations outside of the cell. Finally, we have chloride. Now, chloride is a negatively charged ion, but it is more concentrated outside of the cell. Ultimately, this distribution creates a gradient that drives many physiological processes, which we'll explore further in the next episode on membrane potential. So let's summarize with our key takeaways. Simple diffusion and facilitated diffusion move molecules against their concentration gradients, with facilitated diffusion requiring proteins. And remember, we said to think of those like doors. Active transport moves molecules against their gradient, but it requires ATP. Exocytosis moves large molecules out of the cell, and this also requires energy. Finally, we have osmosis, which is the movement of water driven by solute concentration differences. And along those lines, tonicity describes the effect of solution concentration on cell size. And so that's all for episode 7. I hope you now feel more confident about transport mechanisms and their role in cellular function. Join me next time for episode 8, where we'll discuss membrane potential and how cells use ion gradients to generate electrical signals. Until then, keep questioning, keep exploring, and stay curious.